including corals and sponges.
Yep. Range 30.
A lot of time.
Okay.
That is a sea star.
In the water too, if you notice.
Yeah, the water
there's a lot of
does have
stuff in the water.
Right.
Yeah.
It's a little cloudy looking.
0.2.
Nice sea star.
Good coffee bridge.
Oh, Chris Ma from the Smithsonian. I wonder if he's paying attention on this 4th of July weekend.
Oh, you can see its tube feet. There are those little projections on the underside of the arms.
Mm-hmm.
And I guess modified on the upper side.
Uh-huh.
Another really beautiful lyre or harp. I guess lyre coral. Oh, and a Chrysogorgia. One of the few I can recognize.
They are, much more exciting.
Oh no, they're all exciting. They're all exciting.
To you.
Yeah, go ahead.
Coral is another black coral, the Trisopathes. And then, I see the Chrysogorgia you're talking about. Never mind.
It's another fish in Sirius as you. Or a worm or something.
Yeah, floating around.
Getting kicked around by your heaving.
Another stock sponge coming to center.
Yep.
So a different sponge to the left.
Right, come in as I come by this. This guy, it's not a, is it a sponge coming in?
What do we have here?
Mm.
Oh, look
wow.
at that.
A coiled something.
Well, it looks...
No, it looks coiled, but it isn't, is it?
Well, actually, it looks like those real thick... Oh, it looks like a dead hexactinellate sponge with I guess, maybe hydroids, yeah.
More dead sponges.
Yep.
All right. Graveyard for your sponges.
And a home.
And the home for the colonizers of the dead sponges.
Yep. Oh, give me a home where no sponges roam, and I will make a house for myself.
The way these things kind of like step down.
Mm-hmm. Ooh. I'm losing my stars here. Yeah, I was looking at the flattened but still rounded rocks just below, now behind the ROV, and they, I suspect originally they may have been slabby.
It's a fairly pathetic looking coral.
Indeed.
It's in a good spot. I wonder what happened though. You want to come in tight.
Charlie Brown would be up.
I know.
Well, he would decorate it, and suddenly it would be, you know, a
and
beautiful...
polyps would appear all over it.
Yes. It would be lit up in the center of the gym.
Thanks, pilot.
All right, coming out video.
And Schroeder would play his piano. And Lucy would kick everybody.
It's a little early.
Let's come in on this guy.
Okay.
Happy Independence Day to the U.S.
Lucy would kick everybody.
It's a little early.
Let's come in on this guy.
Okay.
Happy Independence Day to the U.S. We're about 5 hours and 13 minutes before that back in the U.S.
Come on out, Roland.
It's 4th of July out here on the Okeanos Explorer.
Ding,
We're going to have... Yep. We are going to
I
have...
wonder if we could get a look at that large sponge in the center back there.
Yep.
This will be something new. It's
Okay.
very nemated.
Oh, no doubt.
We will do that.
Let's get the fish as
Yeah.
it comes by, and then we'll get the sponge.
Yeah. Thanks, Josh.
Right, come on in, Roland.
Oh, no, you're going to make me spell this name again. I got to look it up.
Copy paste. Copy paste.
True enough.
Got it.
Let that guy go.
Looks.
Hey, Scott, do you think that other fairly pathetic-looking
looking at the
coral that we
concave
saw
side.
had
Nice.
been preyed upon by something? And if so, what would be eating it?
There are asteroids, sea stars that eat
I suspect if
coral.
you get
There
in super
are
close, you might
some
find
kinds
something
of
living on the inside
gastropods,
of
snails, nudibranchs.
sponge.
Okay.
Let's see, sea spiders, although they wouldn't cause the polyps to be removed like that. We'd have to be able to see some predators down here to know what's in this particular area.
Right.
Seems to be a
Okay,
very
we'll go ahead and
strangely
zoom. Thanks,
shaped
Scott.
pink
We'll zoom
thing.
in on this sponge. This looks different from ones that we've seen.
And that white thing to its right also looks different, at least from here.
A white thing to its right?
Pilot ship is in position.
Actually, it almost looked kind of pinkish, almost like.
Yeah, I
I'll
thought.
bet it's another
I saw.
Paragorgia.
Copy that.
On the other
Nav.
side of the rock.
Yeah, maybe. Okay, really nice. Oh, pretty. You can see that fringe of spicules on the edges, and then that beautiful mesh of the spicules and fibers. Chris, any idea what this is? Chris is our glass sponge expert.
Yes, so this is a sponge in the family Pheronematidae, and it's in the genus Poliopogon. And this, believe it or not, is
Now, if you
related
wanna
to
do,
some of those stock sponges that live in the sediment that we've
20
seen a
meters.
few dives back. And you can tell they're similar because you see this base and how it's attached.
1675
This is the lopophytus attachment, or the fibrous attachment of the sponge to the substrate.
And those are our
This
spicules.
particular genus likes to settle on rocks, and not soft sediment. But
This is the full
all
specimen.
of the species that were within this suborder, I think it is Amphidiscophora, all have the same type of attachment.
So what's really interesting to me is that the current looks like it's going like from left to right across your screen. It's really ripping. You can see it at the base, and it really makes me wonder how this sponge that's quite large actually is managing to stay attached to the rock surface with those spicule brushes.
Come on out, Ro.
I think the ends of the spicules, when you look at them microscopically, have little hooks and barbs on them, so they can really hook onto the rock.
Interesting.
Right, come on in.
All right, there you go.
Is this
Looks like another one, like more of the
Bubblegum coral?
bubblegum coral. Is that what you were seeing, Scott, that you thought might have been white?
Yes, that's the one. It
Okay.
looks like what we saw earlier. So
Right.
fine. Another
Okay.
Paragorgiid in the Paragorgiidae family of bubblegum coral.
Great.
This is actually my preferred. This is my coral I like to see. Very colorful.
It is. It's beautiful.
Look at that thing at the
You can see
root.
the smaller bumps on the main stems. Those are called siphonozoids. They're highly reduced polyps that have no tentacles but can channel water in through the colony.
Can I go for
Oh.
full specimen?
All right,
Yeah.
see? I've learned something new.
Thank you, Scott.
Yeah.
Tilt down.
No doubt it won't be the first thing I learned today.
And that is
I mean,
not.
it won't be the last thing I learn, right? It'll be the, yeah.
And
Wanna
that
come
is
out
not
just a little bit?
trivia.
No, it is not trivia.
We're losing
I'll
the whole
also
top of the
point
screen.
out that this one
Yeah,
doesn't
I
have
realize
an
I'm trying to get the
internal
lasers on
axial
it, Roland.
skeleton. Instead, that whole thing is more like cemented together like concrete.
Huh. But it does have sclerites, right? Yeah, sclerites.
Absolutely, it has sclerites, and so the sclerites and the tissue together are kind of glued in there to form all those branches.
So, Scott, sometimes like with, well, a lot of times with sponges, the way the spicules, so the microscopic skeletal elements in sponges, the way they're in the fibers are diagnostic. Do the sclerites arrange themselves in a certain way in these corals, or are they kind of randomly or haphazardly arranged?
You think I'm
And
squishing
it can
the
be
manganese?
both, and that can also be species dependent. So in some species, for example, you'll find a very clearly defined what's called a crown of thorns around the base of where the polyp would retract in. But in something like that Paragorgia, I wouldn't exactly call them random, but they are kind of just sort of jumbled in the tissue, although in some you'll find on the outer, what they call the outer rind, would be one morphological type of sclerite, and then as you get deeper, there'd be another, but they aren't necessarily arranged in any particular way in the bubblegum coral.
Okay, but they might be zoned. So they might be zoned. So you'd see some on the outer rind and some and different ones on the inside. Okay, and this looks like it's very similar to one that was quite common 2 days ago. Stauropathes, is that what this is? Yes, Stauropathes. And you can see the branching, the secondary branching at towards the top of this.
Okay,
Well,
video's
it's
good.
getting tight. Get in tight.
That was tight.
That's all the way.
That's max zoom.
Okay. We can come out.
7.5 in my camera pilot.
ROV nav bridge position move completed.
Thank you, Bridge. Ship is in position.
Copy that, nav.
Is that barnacles on that?
Let's come in on this black stuff right here, Roland. Let's come in right here, Roland. Yep, thank you.
Wow, what is that? What is that? Wait. Okay, there you go.
Thank you. I am wondering what this protrusion from the rock face is. Black and
Like kind of
very thin
tubular. Yeah.
crust sticking up.
Very porous.
Yeah, Diva was
I
saying, is this a dead
haven't-
sponge covered in manganese?
It's
You know,
huge
that's
if
what
it is. I
Well,
mean,
there are some barrel
can there
sponges. I mean,
be?
those of you who dive in, in the tropics, you see these big barrel
Holy
sponges.
smoke!
Oh, heck yeah.
Or cluster of it. Did they come in clusters? Because that could explain why there are
Yeah.
cusps, rounded
Yeah, they
things.
can kind of like.
Okay, maybe
That
that
looks like a
maybe
nice
that is-
specimen.
That's really.
That's amazing.
Sure, what Roland said.
No?
Looks like a nice sample.
Yeah, if you want a lot of manganese with dead spicules in the middle.
Yeah, Diva, I was thinking the exact same thing.
Right, come on out. Let's
Big
get
barrel sponge of some sort. Wow, wow.
Just
Or a
draping that
cluster of
whole
them?
rock.
Yeah,
Yeah, because look,
could
it
very
goes
well
all
be.
the way down that
Yeah.
rock on both sides.
Yeah.
Holy smoke.
For those of you watching, listening at home, those of you who dive in the tropics, especially kind of in the Caribbean, think about those big zestospongia or big barrel
Yes, it
sponges.
is.
Look at the size
look
of
at
this
Layers
one.
on the
this.
rocks,
Oh my gosh.
As well as we come up to it.
Okay, that's one of those
Yeah,
paddle
they're on
sponges,
it.
but a
Ah,
big specimen.
gosh.
Yeah, and there's a fish to
Looks
the
like
right of
Gumby.
it, too. This is a pretty common sponge, so I wonder if we could get
the
that
fish.
fish to the right of it instead.
Maybe. Doesn't crash into a rock.
Nav, if you want to put in the same move, 20 meters, 275, I think that's good.
Yeah, that's good with me.
Copy that. 20 meters, 275.
Poor
2 knots.
thing.
Pilot, watch your port in case you lateral port it off.
Oh, yeah.
That sponge is
you don't
right
wanna
there.
hit that sponge.
Yeah, it's a nice sponge.
Yeah, I don't want to hit that
Bridge,
one.
this is nav.
I don't want to hit any sponges, but I don't want to
Of course,
hit
of
that one.
course.
nav bridge.
Nice.
Same
nice
again,
get shot.
please. Range
Can,
20, bearing
Shirley, can you guys
275,
describe of how old that
8.2 knots.
large sponge may have
Roger
been?
that, ROV nav,
Right, come in on
How
the
old the
head.
large sponge may have been?
275 degrees, 0.2 knots.
Good copy.
First of
Thank you,
all,
Bridge.
I have no idea what kind of sponge it would have been, but I know that some of my colleagues are saying, and I'm not sure I believe this, but some of my colleagues who
Okay, let's
are
get
trying
the.
to
Is that another one? Oh,
figure
okay,
out how
sorry.
old some of these big barrel sponges are that we see in the Caribbean. They're thinking they're hundreds
Let's
of years
push ahead.
old. I'm not so sure about that, but they could be. You never know. I mean,
How-
there's some that are
Nice
like,
big fan here.
2
That's a
meters
beautiful
high
image.
and
If we can get
you know,
that
up to a
just
meter
have a
across.
lot of
Oh,
time.
look how pretty
I get
this is.
7-0. So if you want to do a quick zoom, you can.
Come on in.
Lovely coral.
It's another primnoid.
makes somebody a very busy desktop screensaver. Two mice
2068 meters.
branches at the top.
2300.
Hey, let's look at something really quick.
So,
That
a happy
right there.
day before 4th
Come on
of
in, Ro.
July to those of you on the mainland.
And happy 4th to Guam and
Guam
other
and our
places to the
other
west
territory.
of
Yep.
the
Oh,
date
look
line.
at these. Look at this.
No
See, there's-
leeches.
No leeches. Doesn't look like it anyway. No. We've seen quite a few shrimp with different adaptations. Some are adapted for
Right, let's get the
swimming,
whole guy.
some
'cause
for
I gotta
walking
move.
along the bottom, some
Yeah.
do
Yeah, you're
both.
teetering.
I'm
Some
very
of them are
surprised by
starboard
seeing
side.
how many of these we've imaged in the last few days. Look at all the stalks.
What is that? So
the
sponge
stalks,
stalks, yeah.
So there were a lot of these
Yeah.
really larger.
And now they're covered in
stalks.
manganese.
They are indeed. I don't think that's a lava flow. I think that's a really thick manganese coating on rocks
There
on an
you are.
inclined surface.
And about how long does it take that manganese typically to...
I think actually Chris Kelly was giving us some estimates for how long it takes manganese to form. Chris, I don't remember what you said. You want to take a
Just come in on
take a
this
stab
guy.
at educating me and the rest of our listeners on how long it takes to grow
Sorry,
manganese?
Could you repeat the question again, please?
Yeah, how long does manganese take to grow a millimeter? You were telling me it's like millimeters per thousands of years? I forget.
Yeah, so the average in Hawaii, and this is coming from Dr. David Clegg from the Monterey Bay Aquarium Research Institute, and this is an old paper. He estimated it to be about 2.5 millimeters per million years.
Wow.
And since then, I think we realized that this crust can grow at different rates at different locations, and also the crust itself is supposedly, again, in some places at least, is mediated through bacterial action or something. So it's not just a precipitation. There's some type of biological component to the whole process.
How are we doing on time, Nav?
Okay,
Thank you,
looks like
Chris.
it stopped. Just working it out. We've done 160 meters in the first hour, and we've got about 560 still to do,
geologically, how
and we've got
old,
4 hours
you
left.
guys,
So at this rate, we're doing
estimate
good.
Okay.
Oh,
Copy
well,
that.
you know, in
Thanks,
part,
Nav.
it's
Going.
going to,
Assuming we
the age
go
of the
from
crust
directly
here
from
will
waypoint 2
depend
to the end,
on
and that will
the
depend on you guys
nutrients
and
for
the angle you want to take up the hill.
whatever
Okay,
is
yeah.
mediating the growth. I'm noticing a lot of fractures in the manganese crust, trending uphill.
There is a physical property that affects the growth rate, and that is the level of dissolved oxygen. As it turns out, this manganese crust will precipitate faster at lower oxygen. And so you often find the richest and the best manganese crusts at the same depth as you find the lowest oxygen levels, which is typically between about 800 to 2,000 meters or so.
So, Chris, is that how this site was particularly targeted for potential mining activities?
That's in part, so the depth of the mining activities, that's going to play a role in the decision. But also, these are among the oldest seamounts on the Pacific Plate out here, and that's why there's so much interest in the Western Pacific. And I'm sure that Patty can tell you the story much better than I can, but the Pacific Plate did not even exist about 200 million years ago. It actually came into existence about 185 million years ago. And then it's been moving. It's done a couple bumps and turns on the way, but it's moving gradually toward the northwest. And we are on the northwest side of the Pacific Plate, so we are in the part of it that's the oldest, and we have the oldest seamounts here. And obviously, the older the seamount, the longer time it's had to accumulate manganese crust, and the thicker the crust.
Well said, Chris.
We were just looking at an anemone there. It looked like it had a little tube made of sand grains.
Coral.
I'm just trying to
right
double
of
check
the
and see if it's a cereanthid or not, a tube-forming anemone.
It's the guy on the top left off screen.
The black coral? That guy?
Look at all the stalks.
Sea pen.
Oh,
Right
beautiful.
behind.
Yeah.
Oh, there's a sea pen
And
right in front of the sea pen.
Umbellula. This animal has a bulb right at its base and below the sediment so that it can anchor itself. And on a few of our dives, we've actually seen sea pens that are called rock pens, and you can see that bulb actually attached to the surface of the rock. Another one of the 150 things I've learned on this expedition.
Me too.
Go for it now.
And those dark areas are the pharynx.
Very good, Patty.
Yeah, I tell you, I'm getting
You're
there.
learning way more biology than I am geology, that's for sure. But
Oh, there's just so much more.
Perfect. Thank you, Nav.
And,
Thank you, Bridge.
and the names here in the biological world are much longer,
yes.
harder to remember.
Give you the controls.
Looks like there's a vase sponge to the right edge, halfway up the screen. If we get in close, look straight down the chamber, will be good.
Chris, not sure if this is another euplectellid. These are glass sponges. Glass sponges occur any place deeper than about 200 meters, so we've been finding them on many of our dives.
Yeah, just give me 1 second.
Sometimes you can find them in shallower water and fjords, like in British
still want this,
Columbia
Yeah, if
or
we can like put it right between the skids so we can look straight down.
Yeah,
off the
copy
coast
that.
of Norway. Okay, we're going to look down on this.
Let's make sure we get a look at the base of it.
Okay.
if we could.
Do you want fish first?
So fuzzy.
Now let's take a look at this.
Fish.
All right, we're going to look at the fish first.
Sorry, watchers. I won't move from here.
Okay.
So we'll keep the.
All right.
But this fish is
All right, thanks, Sean.
in a nice position.
They're all kind of shaking their heads.
This guy looks really
to
old.
get rid of.
Yeah, weathered. All right, video. I'm gonna come back down to our first stop.
Okay, I guess before we move in to look straight down at it, they want to look at the base.
Copy that.
Attachment point.
Yeah, Chris, this looks kind of. Oh, this is interesting. This is similar to the one we collected the other day, but it almost looks. See that, like on the outside of it, Chris? It looks like there's like dead skeleton. Is that something that was this? Oh, look, there's kind of like a mesh in between the spicule fringe along the top too.
Yeah, I'm trying to figure out what's going on here too.
Yeah.
The sponge just settled onto a dead skeleton.
Yeah, I know.
Or in the middle of it, or what's happening?
Because it's I don't think it would be in this good shape if it had been regressing.
No,
In the middle.
You know, if it had
Oops, sorry.
dying off.
That was a little quicker.
I'm almost thinking that it's one that settled inside another. Wow, that's very weird. It's very weird, though, because it looks like it's the same kind of the skeleton of the same kind of sponge on the outside of it. Ooh, and there's a little anemone in that crevice also.
Now, what's our heading right now, our bearing?
275.
And slowly tilt back up.
And a polychaete worm tube
Ship's bearing.
as well.
275.
Yeah, that's pretty weird.
Hold.
See hold video?
Yep. See the yellow thing on the upper left.
Do you want to focus on that?
Tina Molotova is calling it a phoenix sponge. It's a new sponge that has risen from the ashes
Yeah, I was saying
of the dead sponge. Oh, I like that,
things as we
Tina.
drift up, like there's a little shrimp there. Also
Tina's a
at the top,
deep sea
so if I
biologist
move up a little
from
more.
the P.P. Shirshov Institute in Moscow. We have another international partner joining us today, Asako Matsumoto from the Chiba Institute of Technology in Japan.
Okay, pilots, keep in
So
position.
through the
Copy that now.
technology that we know as telepresence, we are able to collaborate, partner with scientists from all over the world. They're our shore-based team. We've heard from Chris Kelly and Diva Aman, both from the University of Hawaii. And I think, yes, Les Watling is on the line right now. I think Les is also from Hawaii, as is Patty Fryer. Les is in Maine right now, I think.
You gotta move.
Video
Who else do we have?
quickly, then I have to
Deb
push off,
Glickson
tilt it
from
down.
Harbor Branch Oceanographic Institute,
down.
my colleague at Florida Atlantic University, and the NOAA Cooperative Institute
video
for
got
Ocean Exploration,
5
Research, and
seconds.
Technology.
Just pull off after
We
that.
have
Zoom
Bob
in as you
Carney
go away.
from
4,
Louisiana
3,
State
2,
University
1.
and
I
Scott
have to
Krantz
go.
from the University of
Coming up.
Louisiana. And let's see, on the ship we have joining, and Debbie Blaney. Sorry,
So you
Debbie.
need to push
Debbie
port,
Blaney's
and you
from
need to
NOAA's
push
Office
ahead.
of Ocean
Port and
Exploration and
ahead.
Research, and
Copy
I'm
that. Thank you.
not
And
sure
I'm gonna
where she
pull
is
you
right
up
now,
because I gotta
perhaps
come up.
back in Silver Spring. I
that.
know she was at the University of Hawaii for a
Port
while.
and ahead.
Let's see, Matt Dornbeck is here on the ship with us. He is from NOAA's Deep Sea Coral Research and Technology Program. Matt has been a huge help to Patty and me as we log samples that we've collected, helps us with processing the samples. He's been doing some video work as well, gathering some of the highlight imagery. And down here. There we go.
Okay, the surface is so smooth because of this incredibly thick manganese coating on these rocks. It just
Right
kind
now,
of
if it's getting steep enough,
like
I think
you
we're going to have
are
to slow
laying
down
a
the point.
blanket over a
Kind of
rock
get
plastered
another
over top of it.
surface.
Yeah, put something
Yeah,
over
this
top of
is-
it.
Same again, 275.
Lots
20
of these
meters.
dead
Sure.
Pad point one.
hexactinellid
Copy that.
sponge stalks covered
Yeah,
and
they're
manganese.
This is
very
now
frequent. I wonder if we'll see a huge garden of them
Another
up the
move,
top of
please.
this
This time a
slope.
little bit slower.
I
So
don't know.
range
I think these
20
been here for a
meters.
while.
These
Bearing
have, but
275,
maybe this is a
Yeah,
good place
maybe.
for them.
Maybe.
knots.
And this is just the graveyard.
Spiral stalk coming up.
Good copy.
Above
Thank you.
lasers.
Spiral star.
Wow, look at the collection
Washington,
of
do you
them right
wanna
here in
zoom
this
in?
little
I know.
valley.
All these,
We
this
can
dead
take.
area.
Yeah, we can take a snap zoom if you want.
All right, video. Do you want a quick snap zoom or just yeah?
Gonna fly through some of them.
Yeah, and then we'll go over to the other coral.
Thank you.
All right, that's a good video.
How macabre.
Let's see what's around. Washleads, do you see, is there any interest in right below lasers?
That coiled thing.
Yeah, is there any interest in
I
that?
can take a quick look at it.
Video quick zoom.
It looks like some of those other structures that we saw before. Oh, okay, so we see something
we're looking at
alive.
the
Yes,
coral.
yeah.
And there's an anemone
Maybe.
at the base of it, I think,
Yep, there is.
or below it.
They're drifting up and away.
Yeah.
Losing
And
light.
surely is that
Video.
a carnivorous
Yes, it
sponge?
looks like it may be.
Uh-huh.
Full
Lots
light.
of little things. I'm always impressed by what we see when we zoom in. There's biology all over the surfaces
Push
of this
ahead.
manganese coating on top of the rocks.
Yes, here's some more of those. I'm curious about that spiral structure that we saw. Chris was thinking it was a dead faria that had been then encrusted. Faria is a glass sponge, a hexactinellid sponge, encrusted with manganese. But we're seeing quite a few of these. I mean, not a whole lot, but I've seen probably. Well, we're seeing more now. There's another one over on the left-hand side too. We don't need to get a zoom in on it. I'm just remarking that it just
Copy that.
seems odd. I don't know what that. I don't know what that is.
Mm-hmm. That very first really thick tree-like sponge spickle that we saw had
those like lumps, like
right,
yeah, almost like like mushrooms growing out of a tree or something like that.
Looked
Yeah,
like rungs,
yeah, it did.
you know, because they were hollow in the center.
I know. I don't know what that is. Wait, I should be looking at the chat log because maybe somebody has an idea. No, we're on our own here.
Okay.
Wildly speculating about what
Yeah.
it might be.
Very dangerous.
I know.
Washleads, any interest in dead center?
You want to look at? Oh, so we can go ahead and take a snap zoom on that.
Copy that. This is video
Okay,
snap
quick
zoom,
look.
please.
Quick look.
Thank you.
Oh, do we want to? Scott, do you want to look at that any
That's
longer?
very,
I think.
very delicate.
Is that another Storopathes?
Not a Storopathes, but a black coral, perhaps
Yep.
Trisopathes.
Okay.
You can see black underneath the tissue on the
Trisopathes, yeah.
arms on the
Video.
branches on the left side.
Come back up.
And we're getting a bit more
These are
sediment
weird
here.
corals to look at because it looks like you could almost imagine a polyp has only 2 tentacles, but they don't. They have 6, but they're really stretched out along a branch.
Okay. So, okay, so I'm going to have to take a closer look at that the next time we zoom in on one.
Waypoint, Nev?
After waypoint 2.
Yeah, because it looks like we're
Yeah,
on.
it varies among the
on
black
or
corals,
close to
but in this
waypoint 2.
particular family,
We
the
certainly
polyps
are, yes.
tend to get stretched out in an elongated fashion, so it's very confusing when you're trying to see it in a circular sense because it's actually a
On
thin
the stern of the
rectangle.
ship, we've got about 310, just 308 more or less.
So does science want to go up the cliff until we get to the top
Wow.
And then head over
Pretty
to that
steep
waypoint,
here.
or head directly there from where we are now.
No, we want to go up. We want to go up the top, I think, right?
Go straight
Yep.
up the top.
Yeah,
and
we want to
then-
go straight up the top.
Okay. So we'll just continue straight up at this bearing, or wherever the terrain takes us. And then once we get to the flat, we'll
Go
go
across.
over to that. Yep.
Copy
Gonna
that.
see
Yeah,
something
roger
over
that,
here.
thanks.
Pilot is explaining to us that we
Very
are getting
tall.
some relatively
Yeah,
strong
this is
currents
the same
here.
thing.
So
Video.
we're just talking to him about
Got a
maneuvering.
crab or something at the top.
Yeah. Let me get closer.
There's something on this stock, Shirley.
Crinoid.
I think we're going to pause and take a look.
How do you like the lights video?
Seems a little bit close in the front. You can sort of see in serious view how it's sort of like straight in the front.
Give me 1 second.
So on this
video.
Standing by.
this dead stalk, you can actually see the spicule structures
How's
a
that?
lot better than you
A
could
little
on those
better?
other ones. Those other one. This is one that
Yeah,
hasn't
the top
been
of it is.
encrusted over with manganese yet.
Okay. I'm just trying something new, which is
Looks like there's a
pointing those upper
crinoid
lights
on the
Video
top.
outboard,
man.
so it might be a little less hot right in front of
Yep. Oh, and these have the little curly Q prehensile cirri.
They do
Right.
indeed.
Oh, and
There's a
then there's some
brittle
more of those
star.
branching structures that we saw before, Scott,
Sure.
that we weren't sure what they were.
And a tiny white brittle star at the top. Pretty sure.
So this is a sponge. It's a euplectellid in that family, and it's in the genus Walteria. And those round oscula you see on the sides of it right there, that's very distinctive for something we know as Walteria CF lucardi.
But this one's
And,
dead, right, Chris?
Anyway,
Oh,
this
yeah.
is the first time we've
Pilot,
certainly
looks like ship is in position.
documented, even if it's dead on this dive, and we haven't
Stop
seen
there.
very many at all
Okay,
in
I
the
think we
Marianas
can,
either.
Repeat that move.
Tilt up slightly video.
Copy that.
It's stopped.
Are we all happy with another move? Good.
Yeah, I'll move ahead as soon as we're done here.
What
Till-
are the things, the
continue down.
tentacles?
Tilting down.
I
The base
am
now video?
looking at
Forward,
those
yes.
right
Yeah.
now.
That's a very
I don't
interesting.
know if they
So
are.
must be stoloniferous octocoral.
Yeah.
Joined
So one
together
thing
by
I've
a ribbon
taken
that's probably on the underside
done
of
in the
the
past,
sponge
Sean,
skeleton.
Yes.
You can see eight
is
tentacles.
like
So
when
they're
I'm
definitely octocoral.
Yeah, yeah.
having trouble getting a toe
Bridge.
hold,
This is Nav.
or there's a lot of current.
They grow in the
Yeah.
form of a ribbon or a mat instead of
I'll get it
producing
centered
an
up in
upright
your main
skeleton,
HD,
and therefore
Yeah.
they don't
It's
form
20
the-
and then I'll
meters,
use the
275.
manip cams
Very
That's what I was
figure
trying
out
to do.
Did you see those
how I'm
brittle
moving.
star right in
Yeah.
And I just sort of watch
the
that,
right.
but I have the other thing
The right of the
just in my
crinoid.
peripheral,
Yeah.
so I can see
Nice
if I'm
cracks
like
in the seafloor
drifting
and
left
this
or right or whatever, but I get a
manganese
better
crust.
sense of my
So
motion
there's,
from the
you know,
minute
you
cams.
saw the
Yeah, I
big
agree,
fault
because you
on
get
the
2
top
points of
of the
reference.
seamount.
Yeah,
I
can you
keep
push ahead?
thinking that
Yeah,
there
sorry.
must be
I'm gonna drive
still
right over
some
this guy.
movement.
So
not
partially
a
deployed
lot,
your upper
but
swing
enough to
arm.
start
Yeah, I saw that.
cracking
Does that help
these,
with video?
I'm
these
not sure.
crusts all the way
Copy.
through.
One of the things I've been having trouble with is the near ground is
Really hot.
really hot, but I don't have anything in the background.
Yeah. The only thing I could do with the near ground is turn lights off. So I could turn off the main LED lights, or I could turn off the lower swing arms, for example.
Let's see what happens if you turn off the right swing arm.
I can't turn off a side. I can only do upper or lower.
Oh, okay.
So that's lower off. And that's upper off. So I think the uppers,
Yeah, the upper is definitely
good.
gonna be light in the distance.
yeah, they're filling in those 2 spots on the sides. The lowers, if I turn those on,
Video, can I get a quick zoom? I don't know if this is anything right there.
Sponge.
don't really do a ton.
Sponge.
Is that any
Yeah.
interest swatch rates?
Hexagonalid. No, we're good.
All right, well,
We're
video
good. Thank
now.
you. Probably a
But the
Varied.
right above lasers.
You can see
get
that.
Back in focus. All right, video quick zoom.
Now let's look at.
Any interest watch rates?
Oh, you know, these look similar to those ones we saw underneath the
Yeah,
ledge
the other day.
on a previous dive.
No, that's good. Thank you.
Copy that. Drifting up.
So I'm looking now at some of these stalks, and I'm wondering, are they all sponges or may they have been octocorals? Look at that one that's right next. Yeah, look at that one that's right under the lasers right now.
Copy that video.
Yeah,
The big one on the right.
Yeah, that big one.
Just rotate slightly.
Oh, I see what you mean.
Yeah, look at that. Those look more like
They do.
octocorals than they do sponges.
Yep.
Down to the left.
I'm wondering if all of those were octocorals and not sponge stalks. Wow.
Yeah, that is certainly branching.
I know.
I know
Okay.
you know.
I know.
And you know
What do you think, Scott?
We're exchanging positions here, it seems like, but I feel like that's a sponge.
Really?
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 Person, and I'm the coral person. I think it's a sponge, and you think it's a coral.
Yeah, okay.
That's
It's like
funny.
a treat of flora.
Drifting
So it's the
back.
first time we're seeing any that are, that were more than just the stalk.
Yeah, Shirley, I think the little indentations in the back of this where sediment has collected is giving the appearance of coral branches. That was fooling me for a while, but I think Scott's right. I do think this is a sponge.
But there is a living bamboo coral that was just on the right side of the screen before you zoomed in. Very hard to see.
Okay, yeah, right down at about 5 o'clock, Sean.
Ah, I see it.
See it?
Let me just back up briefly.
That's okay. We'll just make note of it.
No, we're getting it.
Well, look at
This
all
is
the
There we go.
fossilized and manganese-encrusted stalk bases around
I know.
below it.
Video.
So there were big structures there.
Mm-hmm.
Oh, octocoral, definitely.
Rotate down video.
This looks like the one we saw earlier that had the asymmetric distribution of those needle-like sclerites. You can see the 4 prominent white sclerites that seem to be coming out of, on some of these, the lower part of the polyp, but not the upper part. And so this is definitely something that we've seen before.
And you can fly off.
Copy that. Flying off. Pushing ahead.
Now, are all of these black prominences that we see that are maybe 3 centimeters, 3 centimeters high, are the bases of
Dead sponges, maybe?
Sponges?
Yeah, that's what Chris was saying earlier. It looked like they
there
were.
are a lot of them, and it looks.
Another trick, Sean,
I
is
mean,
you'll
they all
notice
seem
that
to
I've
be
been
coated with more or less the
you've
same
looked in
amount
the
of
serial
manganese.
screen. You've
It's
noticed
like
you've
a
been towards the bottom of the
Major
screen a
die-off
lot.
of some sort.
Yeah.
I'm like subconsciously pushing you
So you want to
ahead.
have another really wild guess here?
Ooh,
Very
I
sneaky.
love
I
it.
think I created that move.
Well, it predates
When
you.
this section
That's-
of the
I got that
geo
advice from Todd.
slipped
Yeah, I'm pretty
60
sure you told me
meters,
that advice,
could that
uh,
event have possibly
setting out.
killed a lot of things?
Watch
That's
this. Is this
exactly what I
interest
was
straight
thinking,
ahead?
Chris.
Yeah, let's take a quick look at that, please.
All right, video.
Yeah, I wonder.
Zoom at will.
Oh, what's that? The bottom of
So there's a
yeah.
corollary, but I'll wait till we describe this coral.
Is that worm?
Rotate down slightly.
Still headed forward.
Yeah.
Something wrapped around it.
Yeah.
What is that coiled?
Yeah, I think they're, you have an aplecophoran.
Really?
So we were asking about predators before. This is a worm-like mollusk. Very rarely seen in
Oh.
shallow water. There
Jeff,
are
you
some
wanna
large ones in
you wanna
shallow water,
sit
though,
down?
down in Palau and so on.
This is rarely seen.
Coiled up, it basically
Yeah.
is like a worm. It feeds like
Does give us a better view.
typical gastropods in that it has a radula
Video,
in
do you
there that
want
it can
a sit
scrape
down for
and
this?
we're okay.
Its gills would be at the back end, so presumably the head is at the top of that yellow coil, and back end is lowered down. I'm pretty certain that's what that is.
So you think it's working its way up?
Coral. You can see that below it there are no polyps.
Yep.
All the tissue has been removed, and you only see the skeleton.
Cool.
All right, Washleys, are we good on
We're
this?
good.
or-
You were asking, yeah, I think it's working its way up that
Okay,
that
pilot,
width.
ship is
Yep.
in position.
Looks like it.
In position. Copy that now. Pushing ahead.
You know, this would be an opportunity
Okay.
to
We
can,
to
keep
collect
moving
one
now.
of those
Okay.
Yeah, I
You-
think that's
coffers,
correct.
which I don't know have ever been collected from down here.
Video, did you say
Okay,
something?
hurry.
They're
Yeah.
talking
Are we
about
are we
possibly
able to
collecting.
collect here,
Oh,
or do we have to
Come on.
move?
All right, cancel that ship move.
Yeah.
No.
Copy that.
Copy
Yeah.
that.
Yeah, Washleys, I think we can collect this.
You think it'll stay on? Okay, stand by. You think it'll stay on that stalk, Scott?
Yeah, I don't know. It's kind of coiled up there, so
Okay.
I don't know how rapidly it uncoils. I see Chris has got an ID of Epiminia, and Chris, is that of the apocopheron? Do you think you know what that is? Because if he does, then maybe it's not as critical.
Well, all we know is that the people who specialize in this, and I've forgotten the person's name who contacted me, was pretty sure it was Epiminia, but
Tina.
she didn't give a species name on it.
Okay, wait, sorry to interrupt, Chris, but we're about ready for a ship move, so if we're going to go ahead and collect this, we need to make a decision about it. Sorry, I didn't mean to interrupt, but we need to make a decision about collect or not.
I would wait, but
Well, both Tina and Scott have a really good point here. This is an opportunity to collect this animal that
Copy that,
you wouldn't
Washleys.
otherwise have. They're not that common.
do it.
So I would suggest that we actually give it a go and collect this.
Yeah, so just clip anywhere on that skeleton below the level of the apocopheron.
So you want to collect low?
Yeah.
I'm going to take your camera.
Go for it. Watch it. So we don't think it'll fall off, or we don't know? Okay. Good a snip. All right. I think I'm in a good spot. Don't think I'm moving.
No, you're good.
All right.
Hydraulics are up.
Hydraulics
Do
out.
you know what box you want to go in?
Probably port bio.
Port inner? All right.
Yeah.
Yeah.
I'd say so.
Dry lab, this is co-pilot.
All right. Arm coming live.
Dry lab, co-pilot. Is Casey out there? Yeah, can you grab her to help walk Tara through the sampling?
Arm is live.
Oh, okay. Yeah, dry lab, you can belay that. Sorry, Tara, I was just trying to make sure you had all the help you needed. Let me know if you want a different screen up.
Copy that. Just gonna play with the jaw. That's fine.
What we're doing here is collecting this bamboo coral, but the reason we're collecting this one is because it's got a predator on it, and it's an aplycophoran, so a predatory worm-like mollusk, and it's eating its way up the bamboo coral stalk, and so this will be a good documentation of this predatory behavior, and also probably probably the first sample of an aplycophoran from this area.
Now think about how you're going to grab it, how it's going to be in the jaws when you try to get it in the box. And watch
So I was thinking of
watch your wrist. It's really close to that rock.
Yeah.
I'm just the angle
So
you
don't-
grab
Those
it at.
laser dots that you're saying are 10
Yeah.
centimeters apart,
So I wanted to just
about 4 inches apart.
pick it up, rotate it, and then stick it in.
Yep.
Down. I should be able to do that fine, right?
Yep. Just like if you're grabbing it and it's like this in your jaw,
Yeah.
like this, it's going to be, look at me, it's going to be harder to put in the box than
Yeah.
if you grab it straight up and down. That's all I'm saying. But continue on.
Looks like you got it in, not in the blades at the moment.
Yeah, so I can back that up.
So, I mean, you got it, John.
Yeah. Try this out.
So you're a little close right there. You're going to miss the blades. But
Yeah.
there you go.
Very nice.
I'm going to swap your camera here.
Copy that.
So you're going to have, we can't open the box where you are.
Ah, so I have to lift up.
Yep. So if you want to bring that in front of you, I'm going to tilt your camera.
Actually, I'm going to do this. Alright. Yeah, if you want to do a zoom with video, and then I'll get set up to lift off. Alright, watch it. So I just need to come off bottom, because I can't open my box right here.
Wow, very cool. Okay, that's fine, Sean.
Alright. When you're ready, video.
That's a beautiful image there. That's definitely an
Alright, coming out
aplycophoran,
of drawer.
also called solanogasters. And you can see all the
You might
bamboo
want to,
coral polyps
just 'cause you have to
withdrawn their tentacles. So you can
Come up
Very
anyway.
nicely
You might want
see
to
the
turn to
sclerite.
starboard and put your back into the current.
Turn starboard, put back into current so it doesn't float into me. It'll block the current.
Yeah, it creates a nice pocket so that it won't blow out of the box.
Copy that.
And so you still have auto heading engaged, which is fine.
Thank you.
I mean, just so you know.
Yeah.
But I would just go in auto
Like
depth
auto
and auto
depth.
heading.
Alright, so that pretty much has right there.
Yep.
Auto heading in, and then auto XY.
Yeah, you can try auto XY.
See if I don't drift too much. And Looks like I'm drifting already.
Well, you're just gonna have to go for it.
Yeah. How do you feel in Sirius?
I'm fine.
Okay.
So
I'm gonna go for it.
let me know when you're clear on the box. Ready?
Yeah. If you wanna open.
Box open.
Wow, this is pretty long.
Yeah, and Scott's saying that this whip is not the same as the bamboo
You might need
whips
to
that
go in
we've
the outer
imaged
box.
earlier.
Just because
Yeah,
how
it's
long it
long.
is. Yep. So
Didn't look quite as much.
Yep.
All right.
I see it best in Port Manip.
Tina was saying earlier in the chat that these worm-like creatures even come up in trawls. They don't let go. So, they're pretty tenacious.
Yeah. Food is scarce.
Yeah, for sure.
When you find something.
You hang on.
okay, wrist is on the box.
How do you feel about that?
Same
You're
as
good
out
with
here.
it. Go for it.
You gotta get to lunch early.
And dinner.
And dinner.
You better get to dinner before 5:45 or else it's gone.
A lot of hungry young guys.
All right. Close box, please. I'm clear.
Did it go on?
Yeah, it's that
Outer?
wash rates. Oh, I'm sorry. Yes, outer.
Okay, thanks.
Yeah, it was.
Port.
And it fit. Even though
Outboard.
it's very long.
517.
That was
Special 115. Okay. Very nice. Thank you, Sean.
Very slick
Thank
collection.
you, Pilot Sean.
Oh, anytime. Happy to help.
Okay, that depth was 2015, in case you're wondering.
Shoulder up.
Are my blades on the inside?
It's fine. Shoulder up.
Copy that.
All right, you're good.
Hydraulics off.
Okay.
All right, we can get ship move in. Nav, did you drop a waypoint for that?
Yes, I did.
All right.
All right, I'm just gonna recollect bottom.
Yep.
And we'll be good.
So, same again. 20 meters, 275.1 knots.
Sure. Auto. Yep.
Bridge.
So,
This is now.
Scott and Les and Tina, do you
Oh,
think
I tried
this
to look at you, so I'm turning.
bamboo
yeah.
coral,
Did
I know
you
you
go
So,
Yeah.
range.
Port knob.
noted
20
in the
meters,
chat
bearing
log
275
that
and
it's
speed
different
of
from the one
0.1
we saw
knots, please.
earlier. Is it earlier on this dive? Do you think it's something
you, Bert.
different that we haven't seen at all before on our dives?
I'm not confident that we could say that yet, Shirley, because we didn't actually spend a lot of time looking at the bamboo coral. So I'm not certain. I'll go back and see if I can see any photographs from earlier and from last year to see if I notice anything that I can
Do I
say
need to
is
keep going port, or is that
somewhat
pretty centered?
definitive.
You tell me.
What's the point of having a co-pilot?
He's the captain of this ship anyhow.
Ten minutes in the pilot seat, and he's a tyrant.
Ego has grown astronomically. And that, did you just put a move in? Or was
Yes,
it-
I did.
Oh, okay.
Yes, move's gone. A slow move.
Les Watling
Copy.
and Scott France and Tina Molosova have been
How are we doing on time? How much did that eat in?
chatting in our event